So my team is interested in understanding the molecular and cellular principles of axonal growth. I think to understand why we are asking this question, one has to think at the quantitative aspect of growth. So in our body, we have neurons that can extend axons which are meters long. And I think it's even more, you can see the width of the question even more if you think at larger mammals. So just as a little story, I was one teaching and telling the students, look, think at the giraffe, and so you have motor neurons and sensory neurons. The axons are going to be meters and meters long. And then one student said to me, what about the blue whale? Yeah, it's even worse in the blue whale. It's even a bigger problem. And then you think at extinct animals like dinosaurs, and you think that one cell would extend tens and tens of meters. And so how does this develop during the development of the animal? And then how does this stay and keep functional during the life of a 100-year-old human mammal? So you have this constraint. I mean, this one cell, it has this amazing amount of membrane to keep. And then, even in the brain, even if axons are not, of course, meters long, still you have ramifications of axons. And then, in the end, that means a huge amount, a huge length, a huge amount of membrane. So we're interested in trying to understand how this is made during development and maintained during life, and how it can, in fact, go bad in neurodegenerative disease and in neurodevelopmental diseases. So a lot of our experimental models are with cultured cells, and we also use mouse mutants. But we have, you know, our expertise is in cellular neurobiology, so we focus really on mechanism in neuronal cells.